I'd like to thank Laura Thompson and the FDA for inviting me to participate in this workshop. I'll be talking about statistical approaches for learning about immune biomarker correlates of protection from randomized vaccine efficacy trials. I have in view two major goals of an immune biomarker, which are traditional or provisional approval of a vaccine. And so I'll be talking about statistical approaches to build the evidence base for these types of decisions. And scope of this talk, I'm thinking of randomized placebo-controlled vaccine efficacy trials that use a case cohort or case control design to sample immune markers, one or more, and we're asking the questions, which statistical approaches can be used to quantify evidence for each marker as a correlative risk and protection? Which statistical approaches can be used to compare the quality of a COP across the immune markers? And how to synthesize the results across the different approaches. So I'm going to go through a set of statistical approaches briefly on assessing correlates from a vaccine efficacy trial. And I like to start by dividing statistical approaches into correlates of risk and correlates of protection. Correlates of risk estimate association parameters, things like relative risks or odds ratios, where we're looking to see how does an immune marker correlate with outcome in vaccine recipients. And then correlates of protection analysis is about vaccine efficacy and how does vaccine efficacy somehow causally depend on the immune marker. And the pro of correlative risk analysis is that we can have well-powered assessments. We're doing regression. We know how to do this. We can characterize well how the risk of an outcome depends on a marker or a set of markers. Now, the con is that if we identify a correlative risk, it's not necessarily connected to vaccine efficacy and a correlative protection because of various pitfalls like confounding. And so therefore, we like to also assess COPs directly because they start in the place we want to end up, which is having some data that directly speaks to reasonably likely to predict vaccine efficacy, basically the criterion for at least accelerated approval. And then the con of a COP is that because it's a causal inference method, we need some assumptions that cannot be fully empirically verified. We need more data to get precision. We need sensitivity analysis. So basically, you just need a greater set of data and maybe multiple trials to be able to rigorously demonstrate a COP. So then in practice, I think it's really important to study both cores and COPs. And correlates can be thought about it in two stages. Stage one focuses on, say, the peak antibody time point shortly after the immunizations, and this is where we want to learn about surrogate endpoints. And then stage two is looking at markers over time and looking at so-called outcome proximal correlates, basically looking for what the marker values tend to be around the time of case occurrence. Now, this slide lists different statistical approaches for stage one correlates, and I'll be really focusing on stage one correlates. So first is correlates of risk, as I've mentioned. And then these are different COP approaches. There's the Prentiss surrogate replacement endpoint framework. There's the correlative VE effect modification framework. And then there's different mediation causal methods that haven't seen as much use. And I'll be expressing three different shades of those types of methodology. Now for the stage two outcome proximal correlates, I'll just mention that we can also study correlates of risk by looking at what do the marker levels look like near case occurrence and do some mediation as well. Now, meta-analysis is also important for studying cores and COPs. When we have data that's standardized enough across multiple phase 3 trials, we can look for consistency of correlates across populations and vaccines. We can cross-validate how well a correlate learned from a set of trials applies to another held-out trial. And then we can access meta-analysis surrogate endpoint evaluation methods, which are basically correlating vaccine effects on the marker with clinical vaccine efficacy. So I'm showing an example to rotavirus trials here. And then we've done some analyses of 7 COVID randomized vaccine efficacy trials using binding antibodies and neutralizing antibodies, where here, because we didn't have the same assay to characterize the immune biomarkers, we couldn't do kind of a true correlate analysis. So the x-axis here is using human convalescent sera within each trial as kind of internal control. And it's interesting that even with that caveat, we're able to see these pretty strong correlations that vaccines that had a higher efficacy also had higher NUC titers or binding antibody levels. So this is promising then when we're able to do the more formal correlates analyses, that we'll likely find some correlates. So now I'd like to briefly go through each statistical approach for stage 1 correlates. And I'll be using 2 dengue vaccine efficacy trial data sets for illustration, the SID14 and SID15 data sets. And the vaccinations were administered at month 0, 6, and 12. And then the marker being studied is the neutralizing antibody titer measured at month 13. And the result of the trials was that the efficacy was about 60% in each trial to prevent virologically confirmed dengue of any serotype. And then the efficacy against each of the 4 serotypes ranged from about 35% to 78%. This slide shows the neutralizing antibody marker using the PRNT50 assay. And it's showing data for each trial and placebo vaccine and the cases and controls. That virologically confirmed dengue was the endpoint. And you can basically see that neutralization titers were higher in vaccine than placebo, and they're also higher in controls than cases. So we're starting to see descriptively that there's probably some correlates happening here in this trial. And this slide is summarizing statistical approaches to studying correlates of risk in vaccine recipients. And I think it's important to adjust for baseline exposure prognostic variables in these analyses, because what this is doing is it's trying to make the bridge between core and COP. If we can control for the confounders, then we can causally interpret the regression analyses in terms of immune markers affecting the outcome. And we can use semi-parametric, more flexible parametric, or non-parametric methods to relate an immune marker with the outcome. And I think they all have value. We plan all of them in efficacy trials. And then also when we have a set of markers, I think using a machine learning approach to see how well we can classify vaccinees by the disease outcome, yes or no, or the time to event of that outcome, and looking at different sets of pre-specified markers or pre-specified sets of markers, then we can learn about which markers are best and how can you combine the markers best to predict outcome. This is illustrating one particular non-parametric correlates of risk analysis, which I think of as threshold searching. So this is covariate adjusted threshold searching. So on the x-axis here, we're looking at a subgroup of vaccinees that have the neutralizing antibody titer above the threshold listed on the x-axis. And the y-axis is the risk of dengue, in that threshold subgroup. And a nice thing about these plots is the leftmost point is easy to interpret. That's the overall risk of dengue in all vaccinees. And so then by looking at these curves, you can see how much lower was risk in the threshold subgroups. And especially for SID15, we see that the curve declines precipitously with titer threshold. So that once you get to a titer of about 1,000, 3.0 here, really the risk is about 10-fold lower in that subgroup compared to all vaccinees. So that's pretty good evidence that this is the threshold of very low risk of dengue. Now this slide is showing one way to do machine learning correlates of risk analysis. This is for an HIV vaccine efficacy trial where data were generated on binding antibody markers, on FC effector function markers, and on T-cell markers. And we fit a superlearner model to each of several pre-specified sets of input variables defined by different single or combination of assays. And then we use cross-validated area under C curves to quantify prediction accuracy. And so what we can see from this analysis is you can learn about which markers provide the best prediction and then which combinations of markers provide the best prediction. So here we learned that IgG3 or T cells alone did pretty well. And then if you combine them together, you get a little bit incremental improvement. Now let's turn to COP analysis. So Prentice surrogate endpoint evaluation is very commonly used. Here we're using, to the dengue trials, really a common approach. This is a inverse probability weighted logistic regression where we're estimating absolute dengue risk as a function of titer. We're doing the analysis separately for each of the 4 serotypes. And here for dengue 1, we're seeing that the risk curves are almost superimposed, which is evidence that the Prentice definition of a surrogate endpoint holds. And then the curves are a bit different for dengue 2 through 4, suggesting there's partial surrogate endpoint validity, but it's not perfect. And then for the correlative vaccine efficacy framework, what's done here is we estimate vaccine efficacy over subgroups of vaccinees defined by their immune marker response. And then we get results like here on the right, where we can read off thresholds corresponding to different levels of vaccine efficacy. Now, the challenge with this analysis is that the denominator is the risk of dengue in placebo recipients conditional on what their immune response to vaccine would have been had they gotten vaccine. And this is a counterfactual, so we have to fill it in somehow. And so in practice, we need a baseline variable that is a good predictor of the month 13 NAb titer. And for trials like these Dengvaxia trials, where many people enrolled were previously infected with the pathogen, we can use the neutralizing antibody marker at baseline as the predictor of the immune response, and that works well. And that's why we were able to, I think, have pretty good success in this analysis. And then another approach that is really new, and we have a submitted manuscript on this with Marco and Yuyi, is what we call the controlled vaccine efficacy analysis. This accesses the controlled risk, controlled effects framework of causal inference from Robbins and Greenland in the 90s. And so we're defining controlled vaccine efficacy, where the numerator here is the mean potential disease risk under assignment to vaccine, and assuming we're setting every vaccinee to have the same marker value S. And then the denominator is the disease risk in the placebo group. And then in causal inference, we basically need a known measure confounding assumption, and we need a positivity assumption. And if these assumptions hold, then we can estimate this controlled vaccine efficacy parameter. This slide is showing application of this framework to the Dengvaxia trials, where we happen to use a Cox model to estimate conditional disease risk, and then averaged over those conditional risks to get at our numerator here. And then these graphs here are showing estimation of controlled vaccine efficacy, both assuming no unmeasured confounding and being conservative, that assuming there was some measured confounding that made it harder to infer that the curve varies with the marker. And so the solid curves are the sensitivity analysis that made it harder to find a variation. And you can see that the curves are pretty strongly monotonic in the vaccine efficacy increasing with the marker, even with the conservative approach. So it's suggesting that neutralizing antibodies are a reasonably good correlate protection for virologically confirmed dengue. And then a potential challenge with that method I just mentioned is we have to conceive that we can set each vaccinee to have the same marker level. And this might not be possible. For example, people with certain host genetics or immunocompromised status or advanced age might not be able to achieve the highest immune response. So this method is devised to not assume we can set everyone to have the exact same value of the marker, but rather to specify a shift that everyone starts at whatever value that they have, and then we imagine shifting that upwards, say by a half log or a log, or we could even allow that shift to depend on covariates of the participants. And then we estimate how does the risk of the disease change based on some specified shift, and then how does vaccine efficacy change based on some shift. And then this is showing an analysis of an HIV vaccine efficacy trial where we have T cells, and the result is showing that a shift upwards in the T cell response would not have improved the vaccine efficacy. And then there's classic mediation, which is about natural direct and indirect effects. This is kind of about causal pathway analysis, where for a given marker, we can estimate the proportion of the vaccine efficacy mediated through the marker. And then we can get an estimate of this proportion mediated for each of several markers, and that forms a criterion for ranking them. And these methods haven't been used much in vaccine trials, but they've started to be used, for example, for influenza. This is an analysis of HAI titer for mediating vaccine efficacy against the B. victoriae influenza illness. And in this analysis, Cowling et al., they found a proportion mediated of 57%. So I'd like to conclude by talking about synthesis and interpretation of results obtained from multiple approaches. And the first thing I'd like to say is that some of these analyses, I think, are essentially always of interest to do for any randomized vaccine efficacy trials where we collect the marker data. So we're always going to want to study for each immune marker correlates of risk in the vaccine recipients. And also, if we have multiple markers, I think it's always going to be interesting to do some kind of learning to figure out how do sets of markers classify the outcome, and then to systematically compare different sets of markers for how well they predict the outcome. Now, basically, these first two analyses, by discovering the best correlates of risk, that's going to identify the markers with the greatest plausibility as correlates of protection and surrogate endpoints. And then also, thirdly here, I think meta-analysis surrogate endpoint evaluation is always worth trying when we do have multiple phase 3 trials where we have enough standardization in the endpoints and the immunoassays and other design features of the protocols that it's possible to do a meaningful meta-analysis. I think a meta-analysis is a gold standard approach because we can directly empirically assess how vaccine effects on immune markers correlate with VE. And also meta-analysis is about generalizability and extrapolations, which is kind of at the heart of what we're trying to do in bridging, with accelerated approval, et cetera. We're not going to have direct clinical endpoint vaccine efficacy in the settings we're trying to bridge to, kind of by definition. So then the second synthesis point I'd like to make is that each of the statistical approaches are addressing a different scientific question. So in that sense, they're complementary. And here I'm listing the question addressed for the 5 different stage 1 COP approaches that I mentioned. So the Prentice approach is looking for, do you get a valid inference on vaccine efficacy if you replace the clinical endpoint with the marker? The correlative VE framework is looking for how does VE vary across marker-defined subgroups. So it's kind of a vanilla subgroup effect modification analysis. And then the controlled VE and these other two are mediation ones where we're imagining we can intervene on the marker. And the controlled VE framework is looking for what is VE if everyone is causally set to the marker value. And stochastic interventional VE is similar, except it's asking what is VE if we causally shift the marker distribution in some direction. And then lastly, proportion mediated is about pathways and imagining that if we deactivated the immune response, such that we take a vaccine and we deactivate the immune response to the level it would have been had they gotten placebo, then we ask, is there any vaccine efficacy left? And if the answer is that there's no vaccine efficacy left, then that's a perfect mediation. And so the primary utilities of these different methods, firstly to quantify replacement endpoint validity, then to get signatures of low or high VE, and then for the mediation frameworks, the first two, they're really about bridging and thinking about predicting VE in new settings and getting at mechanism of protection. And then I think in terms of presenting results, I think kind of a consumer reports table where for each of several markers, we do have quantitative metrics of how well they perform on each of these 5 COP approaches. And it's things like how much does the vaccine efficacy curve vary in the marker, or what's the estimate of the proportion of the VE mediated. Okay, and then the details of the study design, the data availability, the amount of data, the assays does affect which approaches are going to apply really well versus might not apply that well. So in general, one can diagnose different parts and figure out which set of methods are appropriate to apply. So all methods are going to apply better if you can collect the major confounders of the effect of the immune marker on the outcome. And then the Prentice surrogate endpoint and the proportion mediated approaches, they'll tend to perform best for vaccines where, well, the studies enrolled people that were previously infected, because they're going to have a lot of variability in the placebo arm, which is really helpful for those methods. Lastly, I find it useful to list the Bradford-Hill criteria for causation and then think about how those map to the utility of the different statistical approaches for learning about correlates of protection. And I think all of these criteria are really important to think about. I'm only going to mention 3 briefly for the sake of time. So there's strength of association. So there, the statistical approaches, very directly give us things like magnitude of regression coefficients and crossfire error into the curve for prediction, and also the COP metrics. On 2 slides ago, I mentioned different metrics that can be used to rank strength of association. Then consistency of association, We can look for replication of results under multiple methods and look at the meta-analysis. And then for biological gradient, we can look for dose response in terms of higher marker values correspond to stronger correlates of risk and stronger correlates of protection. And let me thank my biostatistical colleagues that have been working with me on getting ready for correlates for the different COVID vaccine efficacy trials through the US COVID response team efforts. Thank you.